Hello and welcome to Barkland Labs. This is the video for our fermentation experiment, which will be followed by a distillation of ethanol experiment. I'm going to get this heating because it will take us quite a while. So let's go ahead and turn this on. Okay. So we're just going to let that burn while I explain a few things that we've got going on. So first off, I've got a Erlenmeyer flask full of 200 milliliters of water. One thing I want to point out about that, I've seen some of you guys measuring out water in the Erlenmeyer flask. You never measure straight into this. If you took a really close look, which you can't see, I used exactly 100 milliliters twice from here, poured it in there. And if you look in the line up here, it looks like it's maybe about 210 or so. This is right. That's wrong. These lines are meant to be more of approximations. A graduated cylinder, remember, we went to high school, went to college, that's what you want to trust as far as an actual very precise amount of liquid. So we've got our water heating. I also measured out a few things already. I measured out 50 grams of sugar right here. Sugar is the food that our yeast is going to eat. I also measured out 1 gram of yeast into this paper right here. So each of these weigh papers is just recycled paper. You make a really nice crease there. You can weigh your solid onto the scale, and then it makes it really nice for pouring. You'll see what the pouring looks like in just a bit. So I'm going to let this keep on heating up here. Doesn't really matter what order you add the sugar into your flask. On your instructions, I have you actually adding it once it's hot. Mainly the reason for that, I think, was because I didn't want you guys to burn any sugar just sitting on the bottom. While you are waiting for this, though, you will want to wake up your yeast. Your yeast is very sleepy. So we've got just a small beaker. It doesn't matter what size. It doesn't actually really matter exactly how much water you got in here. I'd say somewhere in the ballpark of 20, 30 milliliters. I've actually got about 35 milliliters in here. You want that water to be lukewarm, and then you want to add a pinch of sugar. So let me run and grab my sugar. Okay, so you add a pinch of sugar. You want to get that a little bit dissolved. And then we're going to add our yeast to that. You can use a stir rod to get that stirring. Sometimes the yeast kind of clings to the side of this little beaker. I don't know if you can see that very well. You want to try to get as much of the yeast dissolved or just in the water, not on the sides of that beaker as possible. What you're doing right now is you're waking up that yeast. In the solid form, it's basically in hibernation, it's dormant. And yeast likes to be dissolved in liquid, and it also likes to have some food. So that little pinch of sugar we put in there, that is kind of enticing that yeast. It's very excited to wake up to a tasty little meal, and it's actually going to start eating the sugar. It'll also start reproducing over time. We're getting it ready so that in another 15, 20 minutes, when we've got our experiment going over here, we can add it to the Erlenmeyer flask, and it's already woken up. We don't want it to try to wake up inside of there. So we're pretty close to boiling now at this point. I'm going to go ahead and pause it and say pretty much when it's at boiling, it's pretty good. There's one step I actually forgot to add in here. I'm not 100% sure I'll keep in. I want you or your classmate to spit into the Erlenmeyer flask. I added that in when I made this lab last year, and I'm not 100% sure I want to keep that. The purpose of that is to introduce bacteria into the experiment, and the purpose was that when you boil it, it's a process called sterilization. You are sterilizing or killing anything that's living in here right now. So I think we might keep that in, but I haven't made a final decision on that. So we're going to turn that off now that we're at boiling. Go ahead and, oh geez. Okay, maybe wait for it to stop boiling so vigorously. There we go. Fun little science lesson as far as what I just did there. Mr. Barkland screwed it up. What I just did there was the water was boiling, and I added a whole bunch of solid granules. And if you remember from the other soda distillation lab, you had to put the boiling stones in there to add more surface area for bubbles to form. And I think what just happened was the water is boiling. We added a bunch of little solid surfaces where extra bubbles could form, and it boiled over. So I'm going to not edit out Mr. Barkland's mistake because it's a learning opportunity. Okay, we're going to go ahead and get that burning again. But I will be reminding you guys that you should wait till it's done doing the vigorous boiling to add your sugar. So you'll notice there's a bunch of sugar kind of just sitting there on the bottom. This is where you want to make sure you get it really nice and well dissolved. Notice that I've got the Bunsen burner heat a little bit lower now. We're going to keep on stirring that. The goal is to really get as much of that sugar, or really all of that sugar, completely dissolved. Okay, we're pretty much there. So now we're going to let it just keep on cooking there for a few minutes. Once again, that is for the sterilization process. If you are an outdoors person, you know that one way that you can clean your water, if you want to drink it, you don't have maybe a water filter, is you can just boil that water, and that should kill any bacteria and ideally any viruses as well that are in there. So we would let that keep on cooking there for a few minutes. For the sake of time, I'm actually going to skip ahead to the final parts of this lab. So fast forward, we've done this for 5 minutes, we turn off the gas, and now you want to get the ice bath ready. Just FYI, notice that your yeast solution should look all frothy now at this point because that yeast is eating. You want to use a hot glove definitely to remove your Erlenmeyer flask from the clamp, and then you are going to cool down the water to an acceptable temperature to add the yeast. So once again, we killed anything that's in there right now, and there's tasty sugars in there. Now we need to put it into the ice bath, cool it down very quickly to a temperature where the yeast is going to want to live in there. So I'm going to grab a hot glove. I'll be right back. Okay, you guys are very lucky. You don't get to see me running around my room grabbing hot gloves. Okay. If you need any help with this, let me know. It's kind of hard. Sometimes these are a little bit unwieldy. The little gloves maybe work better to use one of those, or even just hold it with a hot glove and use the bare hand to quickly turn this. It's usually not too hot. So now we put this in here, and you're going to want to use a thermometer now to monitor the temperature. If you add this yeast too early, you'll actually kill the yeast too. So basically we need to wait until this reaches a point where the water is cool enough that it's habitable, essentially, for the yeast. And that temperature is about 25 degrees Celsius. Let me double check that. Yeah, 25 degrees Celsius is what I've got written on your guys' lab sheet. That's the temperature you're shooting for the yeast. And the term is actually technically called pitching your yeast, and it's kind of a strange name for that. The reason we want it to go quickly is if I just let this sit out on the tabletop and cool down, it would allow just bacteria, other kind of random living creatures, mostly bacteria, that are in the air, possibly contaminate that. So you want to cool it down as fast as possible. Your stopper should also be cleaned. Sometimes you could use something like alcohol to sterilize this, or you could boil this as well. But this is another possible avenue to introduce bacteria to your experiment. So you only want the yeast eating that sugar. Now, while I'm waiting for this to cool down, I'm going to talk about, give you a little idea of what we're doing. So yeast, all these little guys in here, these are microorganisms that like to eat sugar, and we're going to be really mean to those yeast. We're going to let them eat the sugar, but we're going to essentially suffocate them. We're going to cut them off from the oxygen supply that is in the air right now. And many millennia ago, humans discovered that if you cut off that oxygen supply from yeast, they do something kind of funky. They actually still eat that sugar, but they don't eat it as well, and they leave a byproduct behind called ethanol or alcohol. So that process is called fermentation. Once again, it's taking a normal process like eating sugar. That's what we do. It's what yeast does. And then you are cutting off the oxygen supply to make it eat that sugar in a different way. Gives off a different byproduct of ethanol. So I'm gonna keep on waiting. Ideally, I am using my thermometer to measure this temperature. I did not bring my thermometer over here. So I'm going to go ahead and set this down and grab a thermometer. Okay, so we are definitely still above the temperature we need to be at. This is, water takes a long time to heat up. You might have noticed that when you've been cooking pasta. It also takes a very long time to cool down. The further you've got it submerged in the ice, the more that you're kind of stirring it around in there. Those will help it go faster. I'm at 46 degrees right now, so it's going to be a little while. While I am waiting for that to cool down, let me talk about the next part. So, I've got a beaker of bromothymol blue somewhere. Oh.
Attention.
Sorry about this.
Marcella, if you are still on campus, please come to the lower school office. Marcella, if you are still on campus, please come to the lower school office.
Okay, we're going to leave that in on the final edit here. Marcella to the middle school office. So you're going to have a beaker of bromothymol blue. If you remember from our distillation lab, bromothymol blue detects carbon dioxide being produced in your mixture. So what that's going to do for us is that will tell us when the experiment is happening and then when it's actually when it's done happening. So I'm going to grab our bromothymol blue in just a bit. We are almost at our yeast pitching temperature here. We're at 31 degrees. I'm gonna probably pitch it a little bit early. We'll see how that goes. You're gonna have a stopper with an L tube. This is the L tube that you guys are making in our glass bending lab. Part of that is just to really kind of give you an idea that you can make your own chemistry equipment, which I think is kind of cool. And you should have just a single tubing coming out of here. Notice that we don't have any thermometer stoppers in this. You just want one tubing coming out of there. Okay, I think we're probably cool enough now that we can pitch the yeast, or just about there. So I'm going to run and grab our bromothymol blue. I apologize for running off all the time here. Okay, the bromothymol blue. I'm going to keep on letting this cool down a little bit more while I talk. So bromothymol blue is part of our overall experiment. You're going to be putting a stopper on here and putting the end of the tubing into the bromothymol blue. What you've done by setting this little system up here is you have essentially cut off the oxygen supply. Remember when I said we were suffocating the yeast? It's very sad. By putting the end of this tubing into the bromothymol blue, it will allow gas to be pushed out of here and bubbles will be produced, but it's not going to let anything go back into here. So it's kind of a one-way valve essentially. Sometimes we call this a fermentation lock. And then, so that's one purpose, is it's allowing gases come out, that's carbon dioxide, but it's not letting anything come back in, so you're cutting off the oxygen supply. This bromothymol blue also should turn yellow when it's giving off CO2. So a couple days after we get this started, if it's working correctly, you should see your beaker turning yellow. If it doesn't turn yellow, it tells me you've got something wrong with it and you might need to redo it. It'll also turn back to blue when carbon dioxide is not being given off, and that tells us that the reaction is done. This reaction will probably take a couple weeks or so. That's what we're starting at this week, and then we'll leave it on the side of the room and come back to it in a couple weeks. Okay, so I think we are probably cool enough now that we can pitch our yeast, and then we'll be done. So Erlenmeyer flask. Let's give this one last nice little stir. Once again, notice how kind of frothy it is compared to before. That's a really good sign that it's ready to roll. Pour that in there. And then put your stopper on top of there. And now we are good to go. So the yeast is going to start eating the sugar. There's very little oxygen left inside of that Erlenmeyer flask. And then as soon as that oxygen is done, it's going to start producing ethanol instead of just carbon dioxide and water, actually, as the byproducts. And we'll be checking on this over the next couple of weeks, and then finally coming back and distilling whatever our mixture is over here. Thanks for joining me. Have a lovely day. Bye.